So hello everyone, let us begin today's session. I hope my screen is visible now. So hello everyone, let me welcome you all to the week 8 Introduction to Developmental Biology PMRF-NPTEL session. If you have watched the previous videos, you obviously know that PMRF-NPTEL sessions are interaction based problem solving sessions that are taken by Prime Minister Research Fellows such as myself, and we go through the various problems of this particular week's course content and try and solve them interactively with the students that are taking the course. This not only helps the students who are taking the course to be better able to answer the questions of the week's quiz, but also to better understand the various underlying concepts of that week's course content. So to begin with, let me introduce myself. My name is Anuvab Chatterjee and I'm a PMRF Fellow and PhD scholar under the supervision of Professor Jonaki Sen at the Brain Developmental Biology Lab, Department of Biological Sciences and Bioengineering, Indian Institute of Technology, Kanpur. So, to begin with, if you remember, in this particular week's lectures, we have looked at axis specification of embryos, drosophila embryo, and looked into further detail into which genes help in finally making the body plan of the drosophila embryo. So that is what we will be discussing today as well. To start off, let us go through this question first. This is the first question that we will tackle today, and the question asks us: mutations in dash genes cause loss of portions of every other segment, whereas mutations in another blank genes causes defects in every segment. So we during the course of the week, we have seen that there are many genes that are hierarchically activated which help in development of the drosophila axis specification and the body plan. These include the maternal effect genes, followed by the gap genes, the segment polarity, the payroll genes, the segment polarity genes, and finally the Hox genes. So the question is, which genes, if there are mutations in which particular gene, will they be observed loss of portion in every other segment, that is alternate segments, and mutations in which gene will cause defects in every segment? The options are segment polarity and pair rule genes, pair rule genes and gap genes, pair rule and segment polarity, and gap and segment polarity. So I would ask everyone to try and answer. Kavya has also already tried answering it, and she says it is number C. That is, mutations in pair rule genes and segment pair rule genes cause loss of portions of every other segment, whereas mutations in segment polarity genes cause defects in every segment. Okay, so everyone, someone else wants to try and answer this? Okay, so let us move on to the solution slide. So we have seen, so here we have a diagram showing us where exactly which gene is expressed. Pair rule genes, so if we know that there are 14 parasegments present in the drosophila embryo, pair rule genes are expressed in alternate segments. So we will see 7 stripes, as we can see right here. For example, we are here in this example, we are talking about the pair rule gene Fushi-Tarazu, and out of the 14 segments, only Fushi-Tarazu is only expressed in 7, and thus here in the final embryo we will only be able to see 7 stripes of this gene. But if this gene is mutated, then we can see directly that this particular particular part of the segment does not develop. So we only have, so in the final embryo, instead of the bile type having 14 stripes or 14 parasegments, we only see 7 parasegments here. This is because this payroll gene is mutated. Instead, if we look at segment polarity genes, so segment polarity genes are expressed in each parasegment but only one part of it. So one particular segment polarity genes might be expressed just in the anterior part of one parasegment. So we have 14 parasegments. So anterior part of each parasegment means we will be able to see 14 stripes such as this. And we are taking the example of engrail, which is a segment polarity gene. And in the adult embryo or the developing embryo, we will be able to see 14 stripes such as this. And if we have a mutation in the segment polarity genes, this particular portion that where they were expressed, only that will not appear in the final embryo, and thus we have an embryo which looks shorter like this. So in the segment polarity genes case, we have defects in every parasegment, because only the anterior part of it is not present in the final embryo. So we have defects in every segment. Whereas in payroll genes such as Fushitarazu, we do not have that entire segment present in the final embryo. So we have defects present in alternate segments. So here we will be able to see only 7. Here we will be able to see 14, but only one part, either anterior or posterior. And here we have a very nice picture of this right here is probably in situ it is probably an antibody staining against even skipped and Fushitarazu, and we are able to see how exactly. So as I said, Fushitarazu is a and even script as well is a payroll gene, and that is why they are present in alternate segments, so parasegments. So these black stripes, these are each parasegment, we have 7 of those, and red stripes, we have 7 of those as well, which are the other 7 parasegments in which even script is present. So if Fushitarazu was mutated, this entire black parasegments would not be here, and only the red part would have been here. Thus, the answer to the question is payroll genes and segment polarity genes, as Kavya said. Anyone wants to ask anything on this? Okay, so we move on and look at the next question, which is cell-cell signaling in developing drosophila embryo is first seen when? So cell-cell signaling first observed in which stage? When payroll genes begin to be expressed, when segment polarity genes begin to be expressed, when homeotic selector genes begin to be expressed, or when gap genes begin to be expressed. As I said, there is a hierarchy of expression that occurs: maternal effect, followed by gap genes, followed by pair rule, followed by segment polarity, and finally we have Hox genes. What the question is trying to ask us is exactly at what stage does cell signaling play a role? So the options are at the pair rule stage, at the segment polarity stage, at the homeotic stage, or the at the gap gene stage. So this is also a round type of answer, so only one option is correct. Anyone will try and answer this? Kavya says it is number 1 at the stage of pair rule genes. Anyone else wants to try and answer? Okay, so we move on and look at exactly what we are the solution slide. So at the starting, when at the very beginning, the oocyte has a cytoplasmic polarity that we have seen being established in the previous lectures, how exactly via the anterior posterior and the dorsoventral polarity is established via various maternal effect genes, as well as other interaction between somatic and germline proteins such as gherkin and torpedo. And after so the first step is this cytoplasmic polarity being set by maternal effect genes. So here this can show us a gradient of bicoid which is present in the anterior and nano say in the posterior. So this is the first step, the maternal effect genes being the maternal effect genes causing a polarity like asymmetry in the embryo. Next, and we have to remember that at this particular stage, still the zygotic nuclei are transcriptionally inactive. That is, only the maternal mRNAs are only playing a role at this step. In the following step, with starting with the gap genes, little by little the zygotic transcription starts, and various zygotic factors such as zygotic hunchback and various others play a role in order to form this particular gap gene pattern, in which alternate gap genes inhibit each other, and those which are close by help each other to be expressed, and thus we have a big stripe like pattern which is very broad being expressed via the gap genes. Next, the gap genes act in order to help the expression of the payroll genes, and here we have the payroll genes being expressed, and finally then segment polarity genes are being expressed by the help of payroll genes, and all of this together help in the expression of homeotic genes, which actually play a major role in body plan establishment of drosophila embryos. And thus, establishment of expression pattern of maternal effect genes, becoid nanos, such as becoid and nanos, gap genes hunchback, and payroll genes fushitarazu, occur at the syncytial blastoderm stage before cellularization. So one major thing we have to keep in mind is that as this occurs chronologically, so one the first maternal effect, then gap genes, payroll genes, we have to keep in mind that the embryo is also developing a little bit. As time progresses, the embryo is also developing. At the first stage, right about here, the embryo is still a syncytial blastoderm. That is, the nucleus have divided. So we have a lot of nuclei, but they are the cytokinesis has not occurred. That is, cells are not separate. So there is one cytoplasm where various nuclei are floating around. The next step is cellularization of the entire blastoderm, in which individual cells are formed, and then the cytoplasm becomes different for every nucleus. So that is the point when cell-cell signaling will play a role, and that comes after the gap gene stage, as well as the payroll gene stage. Cell signaling is required for expression of the segment polarity gene. So by the time the embryo has evolved to the cellular blastoderm stage, then only can segment polarity genes start being expressed, such as wingless. So this is the stage at which the cell-cell signaling plays a role, because previously there were no separate cells present. Everything was various nuclei that are present on the surface of the entire oocyte, with the same sharing the same cytoplasm. Thus, the cell-cell signaling in developing Drosophila embryos is first seen when segment polarity genes begin to be expressed. So we will be looking at various examples of this in other questions as well. So anyone wants to try ask anything about this? Okay. So let us move on. Next, we have another question. The expression patterns of wingless and engrailed, which are both, let me give you a hint, are payroll genes, are segment polarity genes, are established during which stage? Syncytial blastoderm stage, cellular blastoderm stage, oogenesis stage, or polarization of follicle cells. So, wingless and engrailed, which are segment polarity genes, will be expressed at which stage? Obvious is number 2. And anyone else wants to try and answer this? So as I said, these sessions are interaction based sessions, so I would like everyone to at least try and answering this, because this we just discussed this particular question as well. So expression patterns of wingless and engrailed, which are segment polarity genes, are established during which stage? Syncytial blastoderm stage, cellular blastoderm stage, oogenesis, or polarization of follicle cells? Okay. So since we have just seen it here in this particular slide, cellularization occurs after the payroll genes have been established, and after the cells have been formed, then only cell signaling can occur, and those play a major role in the formation of segment polarity genes. Thus, the expression patterns of wingless and ingrailed, which are segment polarity genes, are established during the cellular blastodome stage. So, we all go on to the next question. Which of the following statements are false with respect to patterning of and axis formation of drosophila embryo? So, which are false is what we have to see. The first option is distribution of different gap gene products overlap. Homeotic genes control the expression of gap genes, payroll genes, and segment polarity genes. Nanos is a maternal effect gene. Nanos is a gap gene. So, which of these are false? So, this is a box type of answer. So, more than one is correct. Kavya says 2 and 4. That is, homeotic genes control the expression of gap genes, payroll genes, and segment polarity genes. And number 4 is nanos is a gap gene. Are false. Anyone else wants to try and answer this? So you can try answering, like, or just typing in the chat whatever you think like might be the correct answer. Okay, so we went a bit too far away. So we were right about here, and this picture is not showing for some reason. So we go back to here as back here. So as we have seen that maternal effect genes include becoid and nanos. So nanos is a maternal effect gene, not a gap gene, so that is false. And the second one is, we have seen that gap genes, payroll genes, and segment polarity genes, all of them play a role in establishment of expression pattern of homeotic genes. But the one of the options was the opposite, that is, homeotic genes play a role in establishment of expression pattern of gap genes, payroll genes, and segment polarity genes. So that is also false. So the option is, and now it is here. The option is, the false options are homeotic genes control the expression of gap genes, payroll genes, and segment polarity genes, and nanos is a gap gene. Nano instead is a maternal effect gene, and these genes play a role in controlling the expression pattern of homeotic genes. And this is also about the distribution of different gap gene products overlap. So in this particular picture where we are seeing these broad stripes, these stripes are not very sharp like the ones we have seen in the parasegment. These can overlap amongst each other, and we see a very fine boundary as well. So we move on. Okay. Once initiated, the expression patterns of ingrailed and wingless are maintained by which of the following? So ingrailed and wingless, as I said, are segment polarity genes. So once their expression is initiated, how is it ensured that which cells maintain those expressions? So the options are activation of Hedgehog production by wingless in ingrailed producing cells, inhibition of Hedgehog production by wingless in ingrailed producing cells, activation of ingrailed production in cells that are already producing ingrailed, activation of Hedgehog by ingrailed in wingless producing cells. So this is a topic that we had touched up upon when we are in the final part of signaling pathways in, I think, 2 weeks back, that we have already seen that we can establish the expression pattern of a particular cell. But how do we ensure that this cell maintains that expression pattern once the signaling is gone? So once the ligands and the signaling that was coming to the cells is removed, how do we ensure that the cell maintains that expression pattern? So that is what is asked. So how do expression patterns of segment polarity genes are maintained? Here we have the example of Ingrain and Wingless, and we have these four options. Options include activation of Hedgehog production by Wingless in ingrain producing cells, inhibition of Hedgehog production by Wingless in ingrain producing cells, activation of engrailed production in cells that are already producing engrailed, and activation of Hedgehog by engrailed in Wingless producing cells. Kavya says number 4, activation of Hedgehog by engrailed in Wingless producing cells. Anyone else wants to try and answer this? So maybe we will go to the solution slide and then try and answer this. So this is what exactly happens in the scenario where Wingless and engrailed are present. What happens is the cells which are producing wingless is obviously a ligand. So it is so this is the exactly this is the Wnt ligand that we had discussed earlier. In drosophila, it is known as wingless. So wingless is produced and secreted out of the cell. The neighboring cell expresses the frizzled receptor, which is the receptor for wingless. And after a signal transduction, this wingless signaling causes transcription of engrailed and another signaling molecule Hedgehog. So we have already looked at Hedgehog pathway. So we have engrailed turning on this in the right cell, as well as production of Hedgehog. Hedgehog is now secreted and it binds to smoothen protein, which is the receptor of Hedgehog. And after another signaling cascade, we have transcription of wingless in the left. So we have a feedback loop that goes on, so that one the left cell only produces wingless and is so wingless is activated in the left cell only, and the right cell only gets engrailed expressed in it, as well as Hedgehog, which goes back and continues the cycle all over again. This is how the expression pattern is maintained. So that only the left cell will continue expressing wingless and the right cell will continue expressing engrailed. So we have to also keep in mind that the beginning of the expression was not due to wingless and engrailed. It was due to the effect of payroll genes. The payroll genes initiated the first transcription of wingless and engrailed in these two, and then this feedback loop continued in order to maintain the expression. So after looking at that, let us again try to answer this question. Once initiated, the expression patterns of engrailed and wingless are maintained by which of the following? Activation of Hedgehog production by wingless in engrailed producing cells, inhibition of Hedgehog production by wingless in engrailed producing cells, activation of engrailed production in cells that are already producing engrailed, activation of Hedgehog by engrailed in wingless producing cells. So anyone wants to try and answer this now? Okay, so is Kavya. Kavya says it is number 1, but I would also like someone else to try and answer this as well. So it like try answering questions. So like even they are wrong, that we can at least have a discussion on them. So yes, it is number 1. As we have seen, it is activation of Hedgehog production by wingless. So Hedgehog production is increased by wingless in engrailed producing cells. So number 1 is the correct option. So we move forward, and the next question is wingless and Hedgehog are. So we have a box type of question. So these are paracrine signaling molecules, these are morphogens, these are gap genes, and product of homeotic genes. Which of the following are correct? Wingless and Hedgehog that we have just seen are the paracrine signaling molecules, morphogens, gap genes, or products of homeotic genes. And this is a box type of answer, so more than one is correct. So anyone wants to try and answer this? Kavya says 1 and 4, they are paracrine signaling molecules and products of homeotic genes. We would like anyone else also to try. So is Sangeeta, are you in the meeting? Hello? Sangeeta? Hello. Probably not. Okay. So if anyone else is online, please try and answering this. Wingless and Hedgehog are paracrine signaling molecules, morphogens, gap genes, or products of homeotic genes. Okay. So we have already seen that Wingless and Hedgehog are signaling molecules that are acting in separate cells, and we know that this is the paracrine signaling pathway in which signaling occurs in nearby cells. So they are already, we know that the first option is correct, paracrine signaling molecules. Second, let us look at this particular diagram. So what exactly is the ultimate like what exactly does this hedgehog and wingless that we have seen production do to the embryo itself? So both of wingless and hedgehog production actually helps in formation of cuticles or small hair-like structures on the embryo itself, and this happens via gradient of hedgehog and wingless. So as we can see right here, as we've seen that if one cell produces wingless, the nearby cell will produce hedgehog, and via inhibition of this, some cells are only competent to produce wingless, some cells are only competent to produce hedgehog. So we see a gradient of hedgehog and a gradient of wingless, just as here. So as you can see, wingless production inhibits hedgehog to come over this side, hedgehog inhibits wingless here, so we have a sharp gradient that falls off in opposite directions. And this gradient ensures different fates in the epidermis, so that different cuticular structures are formed. So we can either have primary, secondary, tertiary, or quaternary structures in which all of them have different natures, and it entirely depends on how the underlying cells, what the gradient of hedgehog and wingless is in the underlying cells. So we have seen already what kind of molecules can do it, in which do this, in which their concentration describes what the reactions of various cells would be. So as we can see, the concentration of hedgehog and the concentration of wingless determine what the phenotype will be of the cells, and this is done by the molecules known as morphogens. So we have seen the morphogens in the previous classes as well, in which we had talked about bicoid and nanos especially, where they help in formation. So highest concentration of bicoid will give rise to the head region and the anterior portion. Highest concentration of nanos will give rise to the posterior portion. Similarly, highest concentration of hedgehog will give rise to primary cuticle and secondary cuticle, and highest wingless will give rise to quaternary ones. So since the expression of various cells depend on the gradient of these hedgehog and wingless molecules, thus hedgehog and wingless are actually act as morphogens in this case. So here we have the figure legend, cell specification of wingless hedgehog signaling center. The first is a bright field image of the wild type drosophila embryo, and second we have a blow up of the A3 section, so that we can look at it at the cuticular structure, and we can see that there are four different nature of cuticular structures being formed. And this particular diagram actually shows us the role of wingless and hedgehog in establishment of this pattern. Either each signal acts in a graded manner, that is the characteristic of morphogens, to specify the fates of cells at a distance from the source. And thus, wingless and hedgehog, in addition of being paracrine signaling molecules, act as morphogens. And they are not products of homeotic genes, since wingless are in real segment polarity genes, and we have discussed that payroll genes help in expression create establishment of expression pattern of the segment polarity genes, and all of them together help in the expression pattern of homeotic genes. So homeotic genes come after. So if anyone does not have any question on this, we move forward. Okay. So we look at the next question. Expression of engrailed is activated by even script. So engrailed is a segment polarity genes, even script is a payroll gene. Although even script is expressed in multiple vertical bands of cells, engrailed is expressed only on one vertical row of cells. This is because, so as we have seen that the expression of even script, since it's a payroll genes, will occur in alternate or the entirety of alternate segments. So we have 7 entire stripes. But engrailed is expressed only in the anterior part of a segment. So if even script which is present in the entire segment, how does engrailed expression be only limited to the anterior part of the segment? The options are: the promoter of engrailed does not bind to even script in other rows. Fushitarazu suppresses engrailed in the other rows. Some other segment polarity genes that suppresses engrailed must be expressed in the other rows, and some other payroll genes that suppress engrailed must be expressed in the other rows. So how does expression of even skip maintain the expression of engrailed, even though the expression pattern of even script is much broader than engrailed? So, and those are the 4 options. Either engrailed does not bind, the promoter of engrailed does not bind to even script in other rows, Fujitarazu suppresses engrailed, other segment polarity genes suppress engrailed in other rows, or other payroll genes suppress engrailed in the expression of other rows. So this is a round type of question. So it is only one type of one option is correct. Anyone try to answer this? Okay, let us look at the solution slide. So we have here a diagram showing us exactly what the segment and parasegments are. So as we have already learned that segments are the morphological character or morphological structures that are present in the embryo. These include the thoracic segments, the abdominal segments, and parasegments are places where the genes are expressed. So these are slightly offset to each other. So we have payroll genes being expressed in this parasegment. So first we have even script, then we have Fushitarazu, again even script, again Fushitarazu, because they are payroll, so they occur in alternate parasegments. What happens is that when there is an the presence of even script Fushitarazu and another payroll genes paired have help in the activation of encrypt. So high concentration of even script or Fushitarazu, and we will have an high expression of engrailed right here. So we have these cells start happily expressing engrailed, but the other genes such as odd skip, runt, and sloppy paired repress engrailed. So we have, so from this portion on, we have an repression of engrailed, so that engrailed expression is only present in these cells, and where both of them are not present, we have wingless expression. Thus, this is a model for transcription of segment polarity genes, engrailed and wingless. Expression of winglets and engrailed is initiated by payroll genes. Engrailed is expressed in cells containing high concentration of either even skipped or Fushitarazu. Wingless is transcribed when neither even skipped or Fushitarazu are active, but there is a third gene such as these odd skipped, runt, sloppy pair, and these help in the wingless expression. And these genes also repress engrailed, so that only the cells at the anterior portion of the parasegment can express engrailed, and no other cells can. And thus we have spatial constriction or restraint on the expression of both, on the expression of payroll genes such as such as engrailed and wingless. Thus the options are: some other segment polarity genes such as runt or odd skip suppress engrailed and must be expressed in the other rows, and thus the expression pattern of engrailed is smaller. So anyone wants to ask anything on this? Okay. So we have been talking about segment polarity genes a lot. Now we move on to homeotic genes. The first option is question is on them. Which of the following is are true about homeotic genes? The options are: one initiated, their expression pattern remains stable during embryogenesis. The characteristic homeodomain is involved in protein-protein interaction. Their 5' to 3' order on the chromosome is collinear. The 5' to 3' order on the chromosome is collinear with their spatial domains of expression in the anterior-posterior direction of the embryo. And the fourth option is they activate the expression of segment polarity genes. So which of the following are correct in case of homeotic genes? Once initiated, their expression pattern remains stable. Their characteristic homeodomain is involved in protein-protein interactions. The 5' to 3' order on the chromosome is collinear with the spatial expression domains of expression in the anterior posterior direction of the embryo, and they activate the expression of segment polarity genes. Kavya says the first option is correct. Anyone else wants to try and answer this? Okay, so let us talk about the Hox genes themselves. So the Hox genes are the genes that are expressed hierarchically finely after their expression pattern is established by the maternal effect, the gap genes, the payroll genes, and the second priority genes all together help in the expression pattern of Hox genes. This gives us an idea that since all of these regulators are involved, Hox genes must be really specially important in their expression, so that all of them together must only help in their expression. Otherwise, they must play a very major role, because their misexpression will cause major defects in the embryo. So the first thing that we have to look into is that Hox genes, the expression of Hox genes is collinear with the in the chromosome with respect to how they are expressed in the embryo itself. So what I mean by that is that, say, Hox genes such as the antennapedia complex, there are the genes labial, DFD, SCR, antennapedia itself. They are expressed in the same order. So labial is expressed first in the embryo, like spatially. I mean, they are expressed first in the anteriormost point, followed by DFD, followed by SCR, followed by antennapedia. And similarly, the bithorax complex of Hox genes are also follow similar expression, similar position on the chromosome. So, since ultrabithorax is present first, it is expressed out just after antennapedia here in the thoracic segment, followed by abdominal A, abdominal B, and abdominal C and abdominal B. So, they are expressed in the later parts of the abdominal segment. So, what I mean is that this is a very nice example of how the position on the chromosome is of the Hox gene is collinear with the expression pattern on the embryo itself, and these are the only genes that follow such a notion. So we had not seen anything like this in the previous genes that were being expressed, such as segment polarity or payroll genes. And these also, there is also the point in the adult fly, we see that they have very restricted expression. So if we lineage trace this particular section, say this thoracic segment, it gives rise to only T2, and it is not expressed either in T1 or T3. And this is also a very important concept that we will learn, that antennapedia itself is very much more important for the development of T2, and if it is misexpressed, it will cause these T2 like characteristics will develop in different part of the embryo, in which we have misexpressed the Hox gene. So Hox genes are very powerful, and they essentially guide which part of the embryo will form what structures, and essentially are the major players in the body plan development of the drosophila embryo. So in this particular scenario, their 5' to 3' order on the chromosome is collinear with the spatial domains of expression in the anterior-posterior direction of the embryo. So number one is not correct. Once initiated, the expression pattern remains stable during embryogenesis. That is not true, because as the embryo develops, its structure will change. So only when Hox genes are being expressed, we can they do not maintain to be expressed in that particular region. So for example, if we are not going to deal with that. So that is the case. So once their expression is established, it does not mean that it will continue to be expressed in that region. But when you lineage trace those cells, you will know that those particular cells only give rise to a particular region. So anyone wants to ask anything on this? Okay. So transformation of a fruit fly mutant into a 4-winged fruit fly is caused by, so as I said, mutations in the Hox gene will cause great changes in the body plan of the embryo. So such mutation is carried out in this particular question, and they are asking if we see a mutant fly which is 4-winged fruit, which is a 4-winged fruit fly. So what mutation has exactly caused this? The options are expression of antennapedia in the third thoracic segment, absence of ultrabithorax expression in the second thoracic segment, absence of antennapedia expression in the third thoracic segment, and expression of ultrabithorax in the third thoracic segment. So in order to better understand this question, we'll go back to this and I will give you another concept. So there is another concept regarding Hox genes. It is known as posterior prevalence. This means that the gene which is being expressed posterior, so say this here we have antennapedia in T2 and ultrabithorax in T3. The gene which is posterior, if that is expressed here, then ultrabithorax will take over the expression of T2 right here. So it will start expressing T3 like characteristics in T2, because essentially the gene which is more posterior will cause which will essentially take over the region where it is being expressed, and this also plays a role in signifying the boundaries of expression. So even the antennapedia might be expressed here. Since ultrabithorax is expressed here, antennapedia's influence cannot cross over. So if an ultrabithorax was inhibited, then only antennapedia can cross over, and this entire region T2 and T3 will take a characteristic of T2 itself. So this is essentially what posterior prevalence means. This particular gene which is expressed posteriorly can overtake the expression of the anteriorly expressed Hox gene. Thus, and here also I am giving another hint that T2 is the one that expresses the wings. So in case we want a fly with 4 wings, what exactly should there be and the options are here. Should there be an expression of antennapedia in the third thoracic segment, absence of ultrabithorax in the second thoracic segment, absence of antennapedia expression in the third thoracic segment, and expression of ultrabithorax in the third thoracic segment? Kavya says 1, expression of antennapedia in the third thoracic segment. Anyone else wants to try and answer this? Okay, so let us go back to the picture itself. As I said, posterior prevalence means that if posterior genes are expressed in the anterior part, they will take over the expression. So if ultrabithorax is expressed in T2, then T2 will assume T3 like characteristics. This also means the opposite way, that if antennapedia is expressed from T2 in T3, it will not have any effect. The reason is ultrabithorax is posterior. So as I said, posterior prevalence means that ultrabithorax, even the antennapedia is expressed in T3, will overtake it. So we cannot have a four-winged fly just by expressing antennapedia in T3 because ultrabithorax will not allow it. What we need is that we need to knock down or mutate ultrabithorax. So if in ultrabithorax is not present here, then antennipedia's expression influence will like antennapedia. So this is another point that is shown here a bit. See how antennapedia is also expressed in these regions as well. So the anterior genes have an expression that spills over to the posterior part. But since there is another posterior Hox gene being expressed there, this expression does not matter at all. Even the antennapedia is already expressed in T3. Since ultrabithorax is there, it will not allow T2-like characteristics of antennapedia to be developed in T3. And thus, the only way we can establish a four-winged fly is that we mutate T3, T3 and ultrabithorax. And once ultrabithorax is removed, antennapedia, since antennapedia is being expressed there, it will take over the T3's usual thoracic segment characteristics and change it to T2. And since T2 forms wings and legs, T3 will also form wings. So we will have a fly that looks like this. So this is a four-winged fruit fly constructed by putting together three mutation and cis regulators of ultrabithorax gene. So cis regulators mean that we know that for expression of a gene there needs to be various DNA elements. So these are enhancers and silencers that are present in addition to the promoter. So enhancers are what determine the tissue specific expression of a particular gene, and since they are present in the same stretch of DNA as the gene itself, they are known as cis. And so this is the same concept as we had seen in organic chemistry. If you had seen the structures, cis means that the same group is present on the same side, trans means they are present on the opposite sides. Similar concept is used here. Cis regulators means that in the stretch of DNA where the gene is present, that particular regulator is also present. So these ordinarily include DNA based regulators or DNA sequences which help in the expression pattern of the gene. These are enhancers and silencers which help. There are other regulators which include trans regulators, which are the transcription factors. So transcription factors are proteins that come from essentially another maybe another chromosome or another DNA segment. They are not present around the gene itself. They are transcribed, translated, and then help in the regulation, and thus they are known as trans regulators. And here in this case, we have mutation in cis regulators. So the enhancers are mutated so that ultrabithorax is not expressed at all. This causes the mutations effectively transform the third thoracic segment into another second thoracic segment. So the antennapedia that was already being expressed is not stopped. Ultrabithorax is removed. So antennapedia transforms the T3 segment into T2-like characteristics and gives an extra set of wings to the fly. So we have 4 set of wings. So this is why we have absence of ultrabithorax expression in the second thoracic segment helps in the expression, helps in this. So is that okay? So we move on, and next question is, which of the following is characteristic of Cartesian coordinate system of gene expression patterns? So Cartesian coordinate system of gene expression patterns, so we mean the spatial x, y in the x, y, z axis. So where exactly a particular gene will be expressed, what maintains that? So in a particular, as we had discussed previously as well, that there are 3 axes present in the embryo: dorsoventral, anterior posterior, and distal medial. These are essentially the x, y, and z axis. So the Cartesian coordinate system of gene expression patterns, where exactly in the spatially a gene will be expressed, is maintained by which of the following? The first option is reciprocal expression pattern as seen in the example of Wingless and Engrailed. Fate of a given cell is determined by the combined effect of gene expression patterns along the dorsoventral and the anterior posterior axes. Ectopic formation of organs, as seen in the case of mutations in antennapedia, and activation of salivary gland formation by SCR at a specific position along the anterior posterior axis. So, which of the following is a characteristic of the spatial or like restricted spatial expression of gene expression patterns? So, this is only one type of answer that is correct. Anyone wants to try and answer this? Okay. So, the option is that fate of the given cell is determined by the combined effect of gene expression patterns along the dorsoventral and the anterior posterior axes. So, say as we have seen that the payroll genes are being expressed in the anterior posterior wise manner. Hog genes are also being expressed in the same way. Similarly, other genes are also being expressed in dorsoventral region. So we have a particular gene expression in stripes in vertical stripes, particularly in horizontal stripes, and a cell will begin expression of further genes when these two stripes combine in a particular place, and that particular cell will have a different cell expression. So essentially, the XY and the X and Y gene expression patterns help in the combined gene expression patterns in the X direction and the Y direction help in fate of a cell being determined. X and Y, as we have seen, is being we call that not X and Y. We say dorsal, ventral, and anterior posterior. So this is the next question, probably the final one, which is consider the following experiments in which bicoid mRNA or a control non-specific mRNA was microinjected into different locations of bicoid mutants or wild type embryos. We have a bicoid mRNA or non-specific mRNA can be the case, and we are injecting it into different location of bicoid mutant or wild type embryos, can be either or. Match the resulting embryonic patterning observed in column A1 to the injection conditions in column 2. So we have a phenotype that we observe here in column 1, and we are trying to match which phenotype actually can be generated via these specific injections. So the first phenotype is acron head thorax abdomen telson, which is the normal one. So acron is the head cap, telson is the final like posterior most structure. So this is the normal embryo. Next we have telson abdomen and telson. That is, it has two tails and no heads. So another like for context, bicoid is necessary for anterior structure. So head, thorax, acron is being established by bicoid. Next, we have in the third telson thorax, and then we have another head and thorax in the middle, and then abdomen and telson. And finally, we have acron, head, thorax, abdomen, thorax, head, and acron. So, a lot of confusing things are there. And the injection conditions are telling us that the first condition is bicoid mRNA is injected, and the anterior of the bicoid minus means this is bicoid mutant embryos. So this embryo does not have any bicoid. Next, bicoid mRNA injected at the middle of the bicoid mutant embryo. Bicoid mRNA injected at the posterior of the bicoid wild type embryo. And injection mix did not have any bicoid mRNA, that is, this was a control. So which of the following options are correct? We have two columns. So anyone will try and match this? Copy says 3. Okay. Okay, so we'll look at that. Anyone else wants to try and match this? Okay. Arul also says it is number 3. Anyone else? Okay, so let us look at this experiment. This experiment was done by Muslim Gulhad and their group, and they actually were the persons who showed us that bicoid and nanos and these are actually act as morphogens, and this was done via elegant set of experiments that we are going to discuss right here. So here first in the left side in the orangish columns we have the controlled conditions, and in the right side we are injecting various mRNAs. So let us go through it. In normal development, bicoid is expressed in the anterior-most portion of the wild type embryo. This we have seen multiple times. Since bicoid is here, it gives rise to anterior structures in a graded manner. So since the maximum concentration is at the top anterior most, it gives rise to acron, followed by a little lower concentration gives rise to the head structures, and a bit lower gives rise to thoracic structures. And after that we have the abdomen and telson as normal. So this is the wild type phenotype. Now let us say at bicoid deficient mutants. In case of bicoid deficient mutants or BCD minus mutants, we have no head structures being formed. Since bicoid was controlling the head structures being formed, we have no head structures being formed, and we have two telsons and abdomen in middle. And the bicoid deficient phenotype is two tails. So can anyone tell me why exactly do we see two tails and abdomen in the middle? Anyone wants to try and answer this? So we had seen that the end structures actually need the expression of the torso. There's another player involved here, which is known as torso-like. So torso-like plus bicoid would have given rise to acron, but since bicoid is not here, we have another tails and right structures, and thus we have two tails and abdomen. So this is the case in case of bicoid deficient embryos, and this is what these are the two stages which we are going to start. The experiment involved taking a bicoid mRNA and adding it to different portion of different embryos and trying to see what exactly the effect occurs. So the first experiment was bicoid was taken, mRNA was taken, and it was injected in the anterior portion of bicoid mutant embryos. So this embryo, in this particular embryo which should have had two tails, we are adding bicoid to the anterior most portion. What happens is this injection essentially rescues the embryo, and we have again normal wild type phenotype of acron, head, tails, thorax, abdomen, and telson being developed. So the anterior structures are developing perfectly. So this shows that bicoid actually has the can actually form the anterior structure and save the embryos, rescue the embryos. So we have normal development. But next, there was also the question, since is only the presence of bicoid necessary and not the is the spatial location of bicoid injection also necessary? So is the only bicoid necessary in the anterior part? So if bicoid was expressed anywhere else, would it also rescue the embryo? So this was answered by adding the bicoid mRNA to the middle of a bicoid mutant. So in the bicoid mutant, if only bicoid was necessary anywhere in the embryo, then we should have a normal development. But instead we saw where the bicoid was injected, we have the head, followed by thorax, followed by abdomen, and then two tails are still being formed. So essentially this does not rescue the embryo but form the anterior structure in the middle. And thus this shows us that bicoid forms anterior structures wherever it is expressed in the embryo. Finally, it was shown via adding bicoid to posterior wild type embryo. So wild type embryos should only have bicoid in the anterior most region. But when bicoid mRNA was injected in the posterior region, it also formed bicoid anterior structure in the posterior. So we have two heads, two acrons, two heads, two thoracic segments, and one abdominal segment. So there are no tail-like structures. And essentially, we have two head-like anterior structures forming on two sides of the embryo. What this shows is that the expression of bicoid should have been in a graded manner. From where the expression is highest, only there the acron will form, followed by head, followed by thorax. And when it is formed in the posterior, injected in the posterior region where there was no bicoid, it also formed the anterior structure in the graded manner right there. So this particular experiment shows us two things. First, that bicoid is necessary for anterior structure formation, bicoid is necessary and sufficient for anterior structure formation, and bicoid acts in a graded manner in order to form this structure. So we have that as bicoid concentration dies down, we have different structures forming. So this proves that bicoid is a morphogen and helps in anterior structure development. So here we have the figure legend. So schematic representation of experiments demonstrating that bicoid gene encodes for the morphogen responsible for head structure in drosophila. The phenotypes of bicoid deficient and wild type embryos are shown in the left here. When bicoid deficient embryos are injected with bicoid mRNA, the point of injection forms the head structures. When the posterior pole of an early cleavage wild type embryo is injected with bicoid mRNA, head structures are formed at both poles. So that is the entire experiment that was done. So anyone has anything to ask from this? This is a very important concept. No one has anything to ask, so I will assume that everyone has understood the experiment perfectly. Okay, so our entire question was essentially this experiment. So we have four options. So first is acron, head, thorax, abdomen, tails. That is exactly the wild type. So we can say that the injection mix did not have any bicoid mRNA, because that is the case. If that is the case, that is only when the normal structures will develop. Second, also we have one with D. Oh no, so no, wait. Injection mix did not have a bicoid mRNA. This will be keep in A. But one can be D, or one can also be A, where bicoid mRNA is injected in the anterior of the bicoid deficient mutant. That is this particular case, bicoid deficient mutant, and we have the injection in the anterior portion. This can also be the case. So we have one can be A or D. Next we have Telson, abdomen, and Telson. So this is again this bicoid deficient mutant, the normal bicoid deficient mutant. So 2 can be D. 2 can be directly D with injection mix did not have any bicoid mRNA. Here they have not written which type of embryo in which they are injected. It will be bicoid deficient embryo. So we have 1A, 2 is D, 3 Telson, thorax, head, thorax, abdomen, Telson. So we have 2 tails and middle portion has head structures. So 2 tails, middle portion has head structure. This is when bicoid mRNA is added to middle of the bicoid deficient embryos. So bicoid mRNA to middle of bicoid deficient embryos, the 3 is B. And finally we have acron, head, thorax, abdomen, thorax, head, acron. So 2 head structures and 2 ends with no tails, abdomen in the middle. That is the final one, in which in wild type embryos, in the posterior part we have injected bicoid mRNA. So we have 2 head-like structure, 1 abdomen, no tail. That was this particular thing. So number 4 goes with C. Bicoid mRNA injected at the posterior end of wild type embryos. That is this particular case. So the correct option that matches all of this is number C. 1 goes with A, 2D, 3B, 4C. Okay. So that was the entirety of what we had to discuss today. We had gone through drosophila axis development, gone through the various genes that are involved and regulate the axis development, and how they are involved in formation of in expression establishment of expression pattern of each other, and finally the body plan genes or the homeotic genes. We have also looked at the last question, in which we have looked into how exactly morphogens act and what experiments were used in order to demonstrate their activity. So if anyone does not, I would ask anyone if anyone has any questions for today's lecture. Okay, so if anyone does not have any questions, we can end right here. I thank you everyone for joining for today's lecture. See you everyone next week. Thank you.